Uh, my talk today is supposed to be number 15th presentation about stem cells. The number one presentation was probably about 20 years ago when I first introduced the concept of stem cells in diabetes to the diabetes caring community in Egypt. And I presented, I gave my first presentation with displaying the new scientific development about the discovery of stem cells which could be taken from embryos, and they were called embryonic stem cells. The stem cells means that the cell which is the original one cell that divides and multiplies for hundreds of times, thousands of times, millions of times, and can go all along increasing its number, and at one time it starts to differentiate and get specialized in different lines to produce different types out of the hundreds of types of cells in the human body. So what you see is embryonic stem cells which are taken from the embryos at particular stage of its development. You take the cells, and these are the cells taken to the laboratory where they can either differentiate and proliferate and increase in number, or take some other pathway, and I will describe this later. Now, one cell starts from the zygote, and at one stage you can take the stem cells which are going either to differentiate into a group of cells which are related to the ectodermal line or the mesoectodermal line. For example, neuron cells are belonging to the ectodermal line, or the mesodermal line, which includes, say, heart cells and muscle cells and blood cells, or the endodermal line, which includes the pancreatic, thyroid, and other endocrine cells, or the germ cells. So one cell, we started by one cell, 2, 4, 8, 16, so on. These are the stem cells, the stem cell, the original cell. Now, what happens? We knew since very long time, since I was a medical student, I knew about stem cells. I knew the term stem cells, but at that time, we knew that stem cells are the stem cells from the bone marrow which produce different types of blood cells. That's all. But now, or later on, we knew that stem cells, which are taken from embryos, embryonic cells, can develop into all different types of cells in the body. Now, for example, if a particular cell is going to stop proliferating and starts to differentiate. أنا بمثلها زي التلميذ بيدخل، دايما كنت بقولها في البتاع، تلميذ يدخل مدرسة ابتدائي وبعدين يروح ثانوي وبعدين يدخل الجامعة، يدخل كلية الطب عشان يبقى طبيب، يدخل كلية الهندسة عشان يبقى مهندس. At one time, the stem cell, a particular stem cell, stops to proliferate and starts to differentiate. For example, an endodermal line of cells have the ability to have high growth potentiality, grows and proliferates. And then it becomes specialized to produce pancreatic cell. And then not all pancreatic, endocrine pancreatic cell. And then beta cell from the pancreas, finally to produce a mature blood cell when it stops multiplication and it starts to function. At this stage, it stops proliferation and it starts to produce or carry on its function. Which means that stem cells can either go into the self-renewal for millions of cells without any hindrance, or starts at one stage to differentiate to become a mature cell, which is either an engineer or a physician or a lawyer, and that's all. It stops differentiating anymore, it stops multiplication. Now, how this happens? A stem cell which is proliferating in the lab, the top, the corner, the right corner up there in the lab. Now, this is the stem cell, and this is the membrane of the blend cells. Through some receptors, it receives a message which either makes it proliferate self-renewal for hundreds, thousands, millions of times without any change, or at one stage by special genetic engineering, etc., I don't go into details of that, it starts to stop this line and goes into that line, and it blocks the self-renewal and starts to function as a specialized cell to produce a particular function. Now, the discovery of embryonic stem cells, which can do this wonderful job of producing different types of cells at different stages, has caused a lot of ethical problems because they were taken from embryos. And I remember before I started to talk about the stem cells in medical conferences, I was lecturing about the ethical aspects of using stem cells in جمعية الطب والقانون, about the ethical problems of using embryonic cells to produce stem cells. Later on, different developments in technique and science developments produced wonderful things during the past 15 years. First we discovered that stem cells can be also obtained from adult bodies. So we can get adult stem cells, which means that we do not anymore need to use embryonic cells, and we can overcome the ethical problems. So we know now that adults can give us stem cells, and this avoids the ethical dilemma and will allow auto-transplantation. Now, furthermore, during the following years, we discovered a treasure of stem cells from the umbilical cord and in the amniotic fluid, were stored, and where these stem cells obtained from these sources can be stored from a fetus to be used for the same person later on in his or her life to produce stem cells from the same body. So there is no problem of rejection, as it could be from any other body. For more details, stem cells which are obtained from the amniotic fluid can contain two types of cells. Some mesenchymal cells, these mesenchymal cells are able to produce two lines. They can produce only one line, mesodermal line only. So they can, when they differentiate these mesenchymal cells which are obtained from the amniotic fluid, we can get from them all types related to the mesodermal line only. But wonderfully, there are also pluripotent. Pluripotent means that they can produce any type of cell. Pluripotent stem cells which can go into the different lines, all the three germ layers. So this is a treasure. You get stem cells from the amniotic fluid and from the umbilical cord without killing any embryos, without going into the dilemma of ethical problems. Now, during the past 15 years, a breakthrough came from the concept or the discovery that after differentiating and finally getting a unified function for a particular cell, you can re-educate this cell to go back to the original stem. What's the problem? I have a problem here. So science has been able to reprogram mature cells اللي بقت مهندس ترجعه من المدرسة الثانوية عشان يدخل كلية الطب عشان يبقى طبيب. So we call this reprogramming mature adult cells back to the stem cell state. Why? In order to be pluripotent and able to produce any other type of cell. بدل ما كان مهندس يطلع طبيب، بدل ما كان محامي يطلع مهندس. How this happens? The production of induced pluripotent cells from mature adult cells which are already specialized, that's reprogramming mature adult cells, was done through genetic engineering, transcription factors with a lot of details. We cannot go into that, but science and technology. Which means that the normal line of producing from embryonic stem cells from embryos to stem cells in the laboratories, then differentiating them to become adult cells differentiated in different types of cells, we can take any of the adult, mature, specialized cell to make it back to the somatic cell stage and becoming pluripotent so that it can start again producing any other type of cell. So it goes back للمدرسة الابتدائي بقى والحضانة وتطلع بدل ما كان مهندس هيطلع محامي. And then after that you allow outside the body the stem cells to proliferate into millions and transplant it back to the patients. Which means that forget about the embryonic stem cells. In another way, this is cartoons that a totipotent cells, pluripotence means able to give different types of cells, multi means that able to give two types of cells, and if it is one type of cell, you can get it back into the pluripotency stage by the scientific development engineering. Now, another breakthrough came during the past years while I was talking about stem cells, is that we knew traditionally that type 1 diabetes is the result of beta cell destruction, and that immune cell reaction leads to this destruction, which means that immune cells are devils which destroy our beta cells, and that proved to be not true. Some of the immune cells are devils, they destroy the beta cells, and some others are angels, which preserve the beta cells. So, in type 1 diabetes, for example, destruction of beta cells results by immune cells through causing inflammation and immune reaction, leading to destruction. These are the devils. But from adult bodies, you can get mesenchymal stem cells, which are the angels. They have potent anti-inflammatory and immune suppression power. So they save the beta cells which are still viable. Which are the angels? Which immune cells are the angels? Without going into details. Some T cells, immune from the immune system, can promote islet cell proliferation. These are subtype, some subtypes, they call T cells, subtype so and so, because they secrete protective cytokines. So forget about the mechanism. These are our angels. But in order to use these angels, send them to the place where they can save the threatened beta cells, you have to go through another process which is complicated, which we call trafficking. Other developments in the research was about trafficking, that sending the angels to the place where they do the rescue process. This trafficking, that's moving the protective adult mesenchymal cells to the threatened pancreatic cells, saves them from immune destruction, which means that this trafficking process has to go through 3 processes. أنا بمثلها بأنه أنا دلوقتي عندي I have a car here in Luxor. I want it to direct it to Cairo rather than go down to Aswan. This is trafficking. Proper trafficking means that you get orientation. أروح القاهرة ولا أروح أسوان. Orientation. Migration. هركب القطر ولا أركب طيارة. And then after arriving to the place, there must be some way to make this cell adhere to the cells which is going to be rescued. Just very quickly, we know the best example of this, the one cell, the spermatozoa, which knows its target, the ovum, in order to fertilize it, and they have the means by the flagella, and they know the way. They get a message from the ovum, they know the way, يسافر للأوفم عارف يروح يلزق فيها على طول عارف السكة تمام. Now we know that since many years. The new developments is that we discovered that in order to send a rescue cell to another cell to rescue it, there must be a signal. From the wound, there is a signal that goes to some other cell away, calling this rescuing cell to go through to this cell. So we start, فاضل دقيقة واحدة ولا دقيقتين. So we need a signal which starts from the اللي بيستنجد. And we already gave you example of the sperms which used by Celia, but we knew with scientific development that a particular cell can repolarize from within its molecules, from inside the cell, in order to get polarization and to get direction تروح القاهرة ولا تروح أسوان, through reorganizing its molecules within the cell. Molecular biology. After arriving to its final destination, it wants to rescue the threatened cell. It has to adhere to it. So we also discovered a lot of scientific knowledge about the so-called adhesive molecules. Because after cell migration, these adhesive molecules are used to make the rescuing cell adhere to the rescued cell. And these molecules are protein molecules. They should be surface molecules in order to attach to the surface of this cell and that cell. And we know now a lot of these molecules, cadherin, integrin, selectin, different examples of cell, and this is which shows you how these adhesive cells work. This is the adhesive molecule which crosses the membrane of this cell. This is intracellular, and this is extracellular, intracellular, the other cell. So these molecules, the adhering molecules, should connect the two cells اللي بتنقذ واللي بتنقذ through these adhering molecules. So a lot of development has been done during the past 20 years, starting from stem cells. And during my first presentation, people asked me, after how long do you think we are going to use stem cells to produce beta cells which can give us? I said not before 5 or 10 years. And we have passed now about more than 20 years, and still we are far from the dream of producing stem cells which can replace the beta cells. I know that about few hundred people all over the world have been successfully treated and stopped taking insulin for 1 month, 2 months, 3 months, 3 years, one case for 8 years. But in general, we are still far from developing the proper success in using stem cells to treat diabetes. Thank you very much.